Let's resume. Now, before going into the break, we were responsible for discussing biomedical waste. And in that, we have discussed that what can be done for this purpose, what steps have been taken in India and elsewhere in the world also for this purpose. So, biomedical waste has been completed. Now, let's complete another type of degradation, and that is called as space degradation. So, we'll try to complete one degradation which is called as space degradation, and also we'll try to cover up what is called as microwave pollution. A question has been asked that, sir, biomedical waste also leads to formation of microbeads. Yes, it can lead to the formation of microbeads. Why? Because biomedical waste, one of the big contents of biomedical waste, component of biomedical waste is also plastic. So decomposition of plastic products would be responsible for what? Would be responsible for generation of microplastics or microbeads. So it can be formed, definitely. You are correct. Now let's talk about another kind of degradation, and this is called as what? This is called as space degradation. See, when a question is asked on degradation in space, it can be attempted in two ways. One is that degradation in space is taking place due to space congestion. Congestion means that more and more artificial satellites today are present in the space. So degradation of space is taking place due to space congestion. Clear? And also we can see that as far as space degradation is concerned, space degradation also takes place. Why? It takes place due to what is called as the space debris. Space debris is also referred to as space junk. Clear? So space degradation also takes place due to the presence of space debris, or which is called as space junk. Now what are these? Now these are debris which are present in the space, and they move at an average speed of 10 kilometers per second. So suppose if they hit or strike the astronauts in the space, instant death would be there. Clear? Instant death would be there. So this is called as what? This is called as space debris. It is also called as I told you that this is also referred to as what? It is also referred to as space junk. So space debris or space junk. Now, space debris can be formed in two ways. One formation of space debris takes place in one way, that is due to it comes from, it can be natural in nature, one can say, because it's coming from the celestial bodies like comets, like meteors. From it would be formed, or it can be formed through anthropogenic activities. Anthropogenic activities means what here? That this can be formed through degradation in space, and that is by we are sending a number of artificial satellites in space. So when these satellites are present in this space, the research equipment, the abandoned satellites may be responsible for formation of space debris. So we can divide space debris into two types. One is natural, and second is what? Second is anthropogenic in nature. Natural space debris, and second would be anthropogenic in nature. So two types of space debris: natural formed through comets or meteors, celestial bodies, one can say. And as far as the anthropogenic space debris are concerned, they would be remnants, or they would be remnant or remaining parts of abandoned satellites, or can be remaining part of also not only abandoned satellites, but also it can be remaining part of the research equipment which we have left in the space. So that would be. Now the biggest problem is extravehicular activity. And extravehicular activity, why? Because when you are talking about extravehicular activity is that activity which is performed in space. Clear? And this is performed beyond the spacecraft. Already we have asked you question earlier in examination on extravehicular activities. So suppose this is your spacecraft, this mobile is your spacecraft, and outside this, when you go outside this, and you are responsible for spacewalk or maintenance of spacecraft or indulging in research, but you are outside your vehicle, spacecraft, then that kind of activity would be called as what? It would be referred to as extravehicular activity. Clear? So that would be called as extra. It can be tethered. Tethered means that I have got out of the spacecraft, but I am connected to it through a wire or a rope. Yes. So that this would be tethered. But if suppose I am not connected with it through a wire or a rope, it would be called as untethered. Untethered. Now, whatever may be the case, the biggest problem in extravehicular activity is what? Is the space debris. Because if suppose the space debris, they collide with the astronaut, they are responsible for striking the astronaut, then certainly what will happen? Then certainly it would be dangerous for them. They can die because at such a speed, instant death would be taking place. But whenever a question on, see, also remember that some of the countries, their acts were responsible for enhancing the amount of space debris. Enhancing the amount of space debris. For example, China blasted one of its satellites in space and said that the satellite was of no use, so it needed to be blasted off. But whatever may the case, it was responsible for creating a vast number of space debris in space. Clear? So that can be said. But whenever a question on space debris is asked, see, space congestion is also enhancing due to 5G communication. And whenever a question on space degradation is asked, we would be answering that with how the methods through which space debris can be removed, or the methods through which space debris can be escaped from. We would be escaping from the space debris. So that would be used in writing the answers. Now, when we talk about methods of removal of space debris, methods of removal of space debris means one method is called as atmospheric drag. Atmospheric drag means, suppose this is the surface of the earth. Let's take the mobile surface of the earth. And the space debris had come within a range of 500 kilometers from the surface of the earth. Here it enters 500 kilometers. So when it enters 500 kilometers from the surface of the earth, it would be dragged by the gravitational force. And when it is dragged by the gravitational force, it enters there, and due to friction, it would be burnt. Friction generated, it would be burnt. So that's called as what? That is referred to as atmospheric drag. That's a natural method of removal of space debris. But second method is called as what? Second method is called as maneuvering. Maneuvering means that we can use a computer model, and this computer model would be determining the time and place of collision, that when it would be colliding with the spacecraft. So the time and place of collision, and with the help of thruster rockets, booster rockets, we can shift the orbit of these spacecraft. Suppose these two are going to collide with each other. So why not shift the orbit of this? So with the help of the thruster rocket, suppose the orbit of the spacecraft is shifted, or maybe this is the spacecraft, it is shifted. So it would be striking, but it would be coming in that direction, but it won't be able to strike because the orbit has been shifted of the spacecraft. So this is called as what? This is called as maneuvering. The second mechanism which was devised was called as shielding. Shielding means that a large metallic shield would be established on the spacecraft, and this metallic shield would be protecting it from the space debris. Clear? Would be protecting it from the space debris. But then you cannot call it a foolproof measure. You cannot say that this is a foolproof measure. Why? Because debris having lesser speed, debris having greater speed can easily, average speed is 10 kilometers per second. Some debris can have greater speed than that. So debris having greater speed can easily penetrate it. And that's why we cannot consider this to be a foolproof measure. Clear? So that cannot be called as a foolproof measure. And the third is what called as, the third would be that we can use mechanical tethers, rope or wires, to lower down this orbit of the space debris. So if suppose the orbit of the space debris is lowered, it would be entering the atmosphere of the Earth, and due to the friction generated, it would be getting burnt. So these are the mechanisms through which space debris can be removed. And when you talk about space degradation, congestion in space has also enhanced due to 5G communication. And more of 5G communication, more and more satellites would be sent to the space. Clear? Right now, space degradation. Let me come to the whiteboard. Space degradation. Space degradation. Degradation. It has taken place. It has taken place due to space congestion. It has taken place due to space congestion. As a number of satellites, is being sent into space, as a number of satellites is being sent into space. As a number of satellites is being sent into space. And with the coming of 5G communication, this number has enhanced. And with the coming of 5G communication, this number has enhanced. This number has enhanced. Clear? Has enhanced. Degradation in space has also taken place due to space debris. Degradation in space has also taken place due to space debris. Which is referred to as space junk, also. And we can see that it is a big threat for what? It is a big threat for extravehicular activity, the activity that you conduct outside your space vehicle or the spacecraft. Clear? So that is called as extravehicular activity. Right on. It is a biggest threat. It is a big threat for extravehicular activities. It is a big threat for extravehicular activities. Clear? Obviously, this is conducted in space. Extravehicular activity. I told you it would be tethered, it would be untethered also. Right, on the average speed at which space debris move, the average speed at which space debris move is 10 kilometers per second. Debris can be broadly of two types. Debris can be broadly of two types. Broadly of two types. Natural. If it is a part of celestial bodies, if it is a part of celestial bodies, like meteors, or comets, like meteor or comets, if it is a part of celestial bodies like meteor or comets. Anthropogenic. If it is a part of anthropogenic, if it is a part of abandoned satellites, or research equipment, or research equipment, abandoned satellites or research equipment, then it would be called as anthropogenic or man-made space debris. Clear? We can see that as far as the natural method of removal of space debris is called as what? It is called as atmospheric drag. Right now, the natural method of removal of space debris, the natural method of removal of space debris, is called atmospheric drag. Is called atmospheric drag. Atmospheric drag. Now that means what? That means that if suppose the debris, atmospheric drag, and what is the meaning of this? If debris reach 500 kilometers within the surface of the earth, then it would be pulled by the gravitational force. So if debris reach 500 kilometers, if debris reach 500 kilometers from the surface of the earth, if debris reach 500 kilometers from the surface of the earth, it would be dragged by the gravitational force. It would be dragged by the gravitational force. So when it enters the atmosphere, due to friction generated, it would be what? It would be destroyed. Right on. When it enters the atmosphere, due to friction generated, when it enters the atmosphere due to friction generated, it would be destroyed. Due to friction generated, it would be destroyed. When it enters the atmosphere due to friction generated, it would be destroyed. Clear? Fine. Now we can see another method for removal of, for escaping from the space debris is called as what? It is called as maneuvering. Right on. Another method. Now that was a natural phenomena. Another method for escaping from debris is maneuvering. And what is this method of maneuvering? What is this method of maneuvering? So computer model would be used in this case to determine the time and place of collision. And then we would be responsible for using what? We would be responsible for using uh, thruster rockets to shift the orbit of the spacecraft. I know a computer model would be used. A computer model would be used to determine the place and time of collision. To determine the place and time of collision. Place and time of collision. of collision. And thruster rocket. And thruster rocket would be used to shift. Would be used to shift the orbit of spacecraft. And thruster rocket would be used to shift the orbit of this spacecraft. Orbit of this spacecraft so as to escape from space debris. So as to escape from space debris. And thruster rocket would be used to shift the orbit of the spacecraft so as to escape from space debris. Escape from space debris. So suppose this is the orbit in which the spacecraft is moving. So with the help of, and this is the debris which is going to collide with it. Debris would be having its own orbit. Spacecraft would be having its own orbit. So we'll shift the orbit of the spacecraft so that there is no collision with the debris. So this method is called as what? This method is referred to as maneuvering. Now the second is the way out is what? That in order to protect the spacecraft, we would be using metallic shields. But this cannot be considered as a foolproof measure. In order to protect the spacecraft, in order to protect the spacecraft, metallic shields in order to protect the spacecraft, metallic shields would also be used. Metallic shields can also be used. Can also be used. Also be used. Also be used. But this is not a foolproof measure. But this is not a foolproof measure. This is not a foolproof measure. And why we are saying so? That it cannot be so secured. We are saying so because that if suppose debris move at a speed of more than 10 kilometers, it can easily penetrate. Down. As debris having greater speed, as debris having greater speed, can easily penetrate it. As debris having greater speed, can easily penetrate it. Can easily penetrate it. Easily penetrate it. Greater speed can easily penetrate it. Is that clear? So that's called as another methods through which we can remove the space debris, and all these other methods through which space debris is removed. One more thing that we can use also the tethers can be used to lower down the orbit of the space debris. Clear? So we can use for that purpose. mechanical teethers, you can say. That's wire or rope. And this would be lowering down the orbit of these space debris. Then mechanical teethers can also be used. Mechanical teethers can also be used, can also be used, also be used to lower down, to lower down, lower down, to lower down the orbit of the space debris, to lower down the orbit of the space debris, space debris, so that it enters the atmosphere of the earth, so that it enters the atmosphere of the earth. And what happens after that, you know very well, that when it enters the atmosphere of the earth, due to friction, it would be destroyed. Clear? So this is space debris that you have to know. Let me see the question that has been asked. Let's see the questions which has been asked. Now, one question has been asked, and that is, sir, how food and water sent to space station? See, one is your international space station, which is always there, which is these days the second most brightest object in the sky during night time after moon. After moon. So this is called as what? This is referred to as the International Space Station, or the ISS. When we talk about how water and food is sent, water and food is definitely required. But first of all, when the astronauts go there, they take it along with them, and then there are assistant missions. Suppose one mission is there already, International Space Station is there, so one mission is sent from Canada, suppose, to assist that mission. It would be carrying the food and this water. Or suppose here also from US another mission is sent, and these would be helping the International Space Station. So sometimes what happens is that those who are present in International Space Station would be brought by these missions, and also new astronauts would be allowed to enter the International Space Station. They are responsible for carrying with them food and water, because without food and water you cannot survive. Clear? Now let's move forward. Last type of question that we have to discuss. One, I'll send you the video of, let me send it also. Why should I delay? I'll send you the video of what is called as invasive species. I think that I have discussed in the class also. But if I haven't discussed, then this video would be fruitful for you. So I'm sending you one video of invasive species into your group. Clear? So apart from what we have studied, look into the invasive species video also. So that's good. Clear? That's good. I've sent it. So after the class, look into this video also of mine, which is dealing with what is invasive species. Because a question on invasive species. Invasive species means what? It means that those species which are not natural to our ecosystem, they're invasive in our ecosystem. They were endemic in some other ecosystem. They either have been brought to our ecosystem or have come automatically. So these species would be called as invasive. That is invasive for our ecosystem. They are endemic in some other ecosystem, naturally found in that ecosystem. But these species are responsible for environmental degradation on a large scale. I've cited some example in that video. One is your Parthenium. Second is what? Second is Lantana. Third is what? Third is the water hyacinth, Christmas bush, Kappa ficus. So all these examples I have cited. So to go through it once, that what is invasive species. And because the question was asked this year also in which species, although the question was very easy, but let's not miss those things. Now, the last form of pollution that we have to discuss, and that's called as microwave pollution. See, we are using mobiles today. In India, we were responsible for converting the pronunciation of mobiles into mobiles. But the correct pronunciation is mobiles only. In India, we use mobiles today. And these mobiles, when they are used, or suppose improper erection of establishment of mobile tower takes place. Clear? When these are used, when improper erection of mobile towers takes place, we can see that due to use of Wi-Fi also, due to all these things, use of mobile, use of Wi-Fi system, and improper erection of mobile towers. What is naturally present in environment today is a frequency, and this is unnatural frequency which is present in the environment today. And this is why, this is because of all these reasons that we have discussed. Use of mobiles on a large scale. Clear? Then also we have discussed that not only improper erection of mobile towers. So all these other factors which are responsible for microwave pollution. See, one of the first report related to microwave pollution was given by IIT Mumbai. Initially, this report was accepted, but later on this report was rejected. Clear? Initially, this report was accepted, but later on this report was rejected. And this report was rejected. Why? See, the team of Stanford University scientists came to India to examine the facts. One or two data were missing. On the basis of this, the complete report was rejected by the scientists of Stanford University. And after that, we can see WHO started to sing the song that no microwave pollution is present. But recently also in WHO office, it was discussed that microwave pollution is present. Irony is what? Irony is that mobile continues to, although WHO says that no micro, initially it said that microwave pollution is present. Today it is saying that no microwave pollution is present. But the irony is what? The irony is that mobiles continue to be in the list of articles according to WHO, which are carcinogenic in nature, which are responsible for causing cancer. So mobile continues to be in the list of articles. If you look at the adverse impact of microwave pollution, the adverse impact of microwave pollution includes almost everything. Almost everything is included in the adverse impact of microwave pollution. Clear? Which includes what? Which includes damage to immune system, damage to nucleic acid. It is also responsible for brain tumors. Incoming call during sleep can alter the brain waves. It has been mentioned. So all these are the adverse impact of microwave pollution. Clear? But see, wherever a question on microwave pollution is asked, there are some species which are linked to it. One is the house sparrows, because the number of house sparrows, their numbers have dwindled, and this is due to microwave pollution. How? How, one can say? Because when you talk about the frequency emitted from mobile towers, the frequency emitted from mobile tower is very less. It's not harmful for us. But then it can prove harmful for the chicks of sparrows which are present inside the egg cell, small ones, which are present inside the egg cell, and due to which their brain can get hampered, ruptured. Clear? So this is the adverse impact of microwave pollution on the house sparrows. See, house sparrows, they share their habitat with you. They would be living where you are living. You are living. So they are responsible for sharing their habitat with you. Clear? First thing about the house sparrows. But their numbers are diminishing. Why? Their numbers are diminishing because one reason for them is microwave pollution, that the frequency emitted from mobile towers. The other reason is what? The other reason is of diminishing number of house sparrows is the urban heat island. Urban heat island means that the entire urban area, due to establishment of concrete structures, has more temperature when compared to those established in the rural areas. More temperature when compared to those established in the rural areas. Just 1 second. See, in rural areas, the temperature is less because the concrete structure is less. So one can say that if suppose this is the urban area, here the temperature is more due to concrete structures. The surrounding rural area would be having low temperature. And that's why this urban area would be called as urban heat island. Clear? Urban heat island. This area is referred to as urban heat island. And due to urban heat island and loss of habitat of sparrows, their numbers have started to reduce. Numbers have started to reduce. Clear? So that's the second reason. Third reason is we use pesticides. We were responsible for using unleaded petrol, not pesticide, unleaded petrol. And unleaded petrol were used in vehicles that it is good, giving you good energy, clean energy. But we can see that for when you are talking about unleaded petrol, when unleaded petrol burns, there is a substance which is emitted. And this substance is called as what? It is called as methyl nitrite. Methyl nitrite. Now, this proves detrimental for the sparrows. Clear? This proves detrimental for the sparrows because methyl nitrite is one of the reasons for their decline. And if you are using pesticides in gardens, that's also not good because pesticides you are using for another purpose. But use of pesticides in gardens is responsible for restricting the food reservoir of house sparrows, because of which their number can dwindle. So all these are the reasons behind dwindling number of house sparrows, and which is directly linked to what? Microwave pollution. First of all, let's see this. Just coming in a few seconds. See, due to diminishing number of house sparrow, an effort was made by ONGC, and this was made in the year 2007, which is called as Save Sparrow campaign. Save Sparrow campaign means that we would be re-establishing the habitat of house sparrows. So this is called as Save Sparrow campaign. So this was made by, and this proved to be fruitful also. Clear? Another thing which comes into picture is what? Honeybees. See, there's a behavior among insect which is called as what? Which is called as swarm behavior. Swarm behavior. And swarm behavior means that the insect would be interacting among themselves. See, in which sense you are trying to ask me how centripetal and centrifugal force influence space debris directly or indirectly? That is only one way affected, and that is the movement of the space debris is affected due to these forces. Next thing, see, there's a behavior among insect which is called as swarm behavior. Swarm behavior means that the insect would be interacting among themselves, and they also interact with environment. So this type of behavior is called as what? This type of behavior is called as swarm behavior. And as far as swarm behavior is concerned, honeybee is also an insect. It would be having swarm behavior. Now, due to microwave pollution, it is said that the swarm behavior of honeybee gets disrupted. And if the swarm behavior of honeybee is getting disrupted, it means that the habitat of honeybees would be also affected. So that's another adverse impact of microwave pollution. We can see with the coming of 5G communication, the health experts have said that the radiation level would be enhancing. So there's a growing concern that the radiation level would enhance due to microwave pollution, and this is said by the health experts. Health experts are responsible for saying so. But as far as 5G communication is concerned, 5G communication can be justified also. 5G communication is essential for Industrial Revolution 4.0, which the government of India has launched, because this is based on artificial intelligence and this is based on machine-to-machine communication, for which 5G communication would be certainly good. But for 5G communication to be implemented worldwide, more than 20,000 satellites would be sent into space. This would be enhancing space congestion. That's why we are concerned about 5G communication. And when you send these satellites into space, you are sending them through rockets. You are responsible for sending these satellites through rockets. So they would be emitting black carbon, they would be emitting blue carbon. So that's another concern for us. Clear? Write down microwave pollution. Microwave pollution. I use the white word now. Microwave pollution. Pollution. Due to, write down the topic, microwave pollution. Microwave pollution. due to microwave pollution due to use of Wi-Fi system, mobiles, use of Wi-Fi system, mobiles, and improper establishment of mobile towers. Improper establishment of mobile towers. An unnatural frequency. natural frequency. Frequency. An unnatural frequency is constantly present in the environment. Is constantly present in the environment. Unnatural frequency is constantly present in the environment. Which is termed as, which is termed as, termed as which is termed as microwave pollution. Which is termed as microwave pollution. Microwave pollution. Clear? And we can see that one of the first reports related to it was given by IIT Mumbai. Write down, one of the first reports related to it was given by IIT Mumbai. Given by IIT Mumbai. WHO initially accepted this report. WHO initially accepted this report. Initially accepted this report, but later on rejected it. Initially accepted this report, but later on rejected it. Later on rejected it. But the irony is what? The irony is that mobiles continue to be in the list of articles according to WHO, which is carcinogenic in nature. Right on. But mobiles continue to be mobiles continue to be in the list of articles. Mobiles continue to be in the list of articles according to WHO. According to WHO, which is carcinogenic in nature. Which is carcinogenic in nature. Carcinogenic in nature. Carcinogenic means that it would be responsible for causing cancer. Carcinogenic in nature. Fine. And as far as the adverse impact of microwave pollution is concerned, the adverse impact of microwave pollution would be including many. See, up till now, they haven't asked you question on microwave pollution, space degradation, or also biomedical waste. These are the potential areas from where the questions can be framed. Right now, the adverse impact of microwave pollution includes the adverse impact of microwave pollution includes the adverse impact of microwave pollution includes damage to immune system, damage to immune system, damage to nucleic acid, damage to immune system, damage to nucleic acid, nucleic acid, nucleic acid, then brain tumor, brain tumor, brain tumor, Then also alteration of brain waves. And alteration of brain waves means that if suppose a person is sleeping with his mobile set on, the incoming call can alter the brain waves. Incoming call can alter the brain waves. So that's the alteration of brain waves that we are talking about. Fine. So brain tumor, alteration of brain waves. But it's not only this. Microwave pollution is also responsible for what? Microwave pollution is also responsible for. Also responsible for. Just one second. Alteration of brain waves. But see, apart from the impact that microwave pollution has on humans, it also has impact on birds and honeybees, insects. So microwave pollution is responsible for what? Microwave pollution is responsible for the uh, diminishing number of house sparrows. Right now, this pollution is also responsible for. This pollution is also responsible for diminishing number of diminishing number of house sparrows. Diminishing number of house sparrows. Also responsible for diminishing number of house sparrows. How? See, the frequency which is emitted from mobile towers has a thermal impact. Right on. The frequency emitted from mobile towers, the frequency emitted from mobile towers, frequency emitted from mobile towers has a thermal impact, has a thermal impact on the chicks of sparrows, on the chicks of sparrows. That's the newborn ones, chicks of sparrows. And these are here in this case, it is not born even, it is present inside the egg cell. Chicks of sparrows present inside the egg cell. Chicks of sparrows present inside the egg cell. Present inside the egg cell. Due to which their brain gets ruptured, due to which their brain gets ruptured. So suppose chicks' brain get ruptured, how they would be born live? They are dead. So that's the connection between microwave pollution and diminishing number of house sparrows. But it does not mean only this. It also means that the diminishing of house sparrows is not only due to this. The diminishing number of house sparrows is due to other factor also. Right on. The number of sparrows have reduced also due to other factors. The number of sparrows have reduced also due to other factors. Sparrows have reduced also due to other factors. Also due to other factors. Which includes also due to other factors, which includes the number of sparrows have reduced also due to other factors, which includes first, and not only this, there are other additional factors. First, use of pesticides in gardens. In gardens. And we are responsible for using them. Now this restricts their food reservoir, right on, which restricts their food reservoir, which restricts their food reservoir. Their food reservoir, which restricts their food reservoir. Now that's one. But also we can see not only use of pesticide in gardens, but also we can see that use of unleaded petrol is also responsible for it. Right on. Use of unleaded petrol was also, we used it earlier, now we have phased it out. Use of unleaded petrol is also responsible for it, is also responsible for it. As combustion of it is responsible for. Combustion of it means combustion of the unleaded petrol. As combustion of it is responsible for. Combustion of it is responsible for emission of methyl nitrite. Emission of methyl nitrite. Responsible for emission of methyl nitrite. Methyl nitrite, which is detrimental for sparrows. Which is detrimental for sparrows. Detrimental for sparrows. Detrimental for sparrows. Clear? Detrimental for sparrows. And also one can say that the establishment of urban heat island is also responsible for it, because when urban heat island is established, the habitat of house sparrows would be reduced, because you are responsible for establishing concrete jungles. So the temperature of the urban area would be enhancing, but in that of the rural areas would be less. Establishment of urban heat island. Establishment of urban heat island is also responsible for it. Establishment of urban heat island is also responsible for it. Is also responsible for it. As the habitat of house sparrows. As the habitat of house sparrows gets affected. As the habitat of house sparrows gets affected. Gets affected. Clear? And do remember that microwave pollution is also responsible for disrupting the habitat of honeybees, because its swarm behavior gets affected. Swarm behavior means the interaction among themselves and the interaction with environment. So you can see the swarm behavior gets affected, and that's why we are saying that the question asked is, sir, is wireless headphone and digital also generate microwave? Yes, it generates. Yes, although it's less harmful for us, but it definitely generates. That's why wired items are better. Urban heat island is also responsible for disrupting urban heat island is responsible for disrupting the habitat of house sparrow. That you have written. That you have written. No need of repeating it again. But remember that microwave pollution is also responsible for disrupting the habitat of honeybees as their swarm behavior gets affected. Right on. Microwave pollution is also responsible for microwave pollution is also responsible for disrupting disrupting the habitat of honeybees, disrupting the habitat of honeybees as their swarm behavior as their swarm behavior also gets disrupted as their swarm behavior also gets disrupted. Swarm behavior gets disrupted, not also, as their swarm behavior gets disrupted. Clear? You would have seen ants moving in one row and some ants joining the row, bringing food stuff from other part. So this behavior is called as what? This is called as swarm behavior. So this swarm behavior also gets disrupted because of the pollution. Clear? Fine. Microwave pollution. And with the coming of 5G communication, we can see that microwave pollution would be enhancing as the radiation level would be enhancing. Health experts have warned. Right now, with the coming of 5G communication, with coming of 5G communication, health experts have warned against radiation risk. Health experts have warned against radiation risk. Radiation risk. And why we are saying radiation risk? Because definitely the radiation would be enhancing. Radiation risk. Clear? So definitely the radiation would be enhancing. So why should this radiation, we should not take this radiation risk according to the health experts. But then 5G communication is justified also. Why? Because India has launched Industrial Revolution 4.0, which is based on artificial intelligence and machine-to-machine communication. Right? But 5G communication is justified also. But 5G communication is justified also. Justified also. As India has launched Industrial Revolution, as India has launched Industrial Revolution 4.0, as India has launched Industrial Revolution 4.0, which is based on machine to machine communication, which is based on machine to machine communication, based on machine to machine communication and artificial intelligence, and artificial intelligence, and artificial intelligence. Clear? This much in microwave pollution. This brings to the end of the chapter pollution. Clear? This is bringing us to the end of the chapter pollution. So this much completed. One portion, big portion. Fine. So one big portion has been completed. Now let's take a pause of 2-3 minutes and then we start with another topic. Change your copies also, and that is biotechnology. Clear? So let's start with biotechnology and let's look into the types of questions which are asked in our examination. Let's take a break of 2-3 minutes and then we start, because get your new copy also, because one environment portion you have completed, only one portion is left, and that is disaster management, which you will do after completing biotechnology. So first of all start with biotechnology. So just take a break of 2-3 minutes. Let's start. Now, see, what are the questions that you have to attempt? First of all, you have to know about biotechnology. In biotechnology, you have to cover up many topics. One topic is called as human genome, and also DNA fingerprinting. Are there exceptions to DNA fingerprinting also? So is there any exception to DNA fingerprinting? Can a person have more than one DNA fingerprint? That needs to be also covered up. Then we have to cover up what is called as stem cells. So let's cover up stem cells also. Fine. And then we'll cover up what is called as cloning. Triple parent baby needs to be completed. Fine. Triple parent baby needs to be completed also. So that is also to be covered up. Then we'll cover up also designer babies, CRISPR-Cas9 technology, GM crops, all these needs to be covered up in biotechnology. So let's start and discuss biotechnology. Now, for this purpose, we would be using the board because orally biotechnology cannot be discussed. So for this purpose, we will be using the board. And let's start from the board itself, because without board, it cannot be discussed. Each and every part relates to the board itself. So let's discuss that. Let's start. Clear? So let's go to the board first of all. See, when we talk about biotechnology, we can see that first of all, we have to know, first topic that we would be covering up is called as what is human genome. But to understand that, first of all, let's draw a generalized cell. Generalized cell means, when we are talking about generalized cell, it means that it would be having the characteristic of both plant cell and animal cell. Clear? So it has a characteristic of both plant cell and animal cell. This is the nucleus in a generalized cell. And inside the nucleus, you can see bearers of hereditary characteristic is present. Now, this bearers of hereditary characteristic would be called as what? It would be called as chromosomes. Clear? Chromosomes. And here we can see, suppose in plant cells, we know that plastid is present. Here, suppose in animal and plant cell both, mitochondria is present. So this is a generalized cell. When you talk about chromes, when you talk about generalized cell, in the generalized cell in the nucleus, we can see that chromosomes are present. Chromosomes are what? Chromosomes are bearers of hereditary characteristic. They are bearers of hereditary characteristic. And these chromosomes are made up of DNA. So they are constituted by DNA. So suppose we say chromosome is a molecule, then DNA would be its atom. So they are made up of DNA only. And DNA is not only confined to the nucleus of the cell. It's not only present in the nucleus of the cell. Clear? DNA, which is mainly present in the nucleus, but it's also present in two other parts. One is mitochondria and second is called as plastid. Mitochondria and plastid. Now, these kind of DNA which are present in mitochondria and plastid, they are referred to as what? They are referred to as extra chromosomal DNA. Extra chromosomal DNA means that they are present beyond the chromosomes. So they would be referred to as extra chromosomal DNA. They are present beyond the chromosomes. Clear? So this is about mitochondria and plastids. And then the question is, what is genes? Genes are nothing but parts of DNA only, which would be responsible for reflecting different kinds of your characteristic. So they would be referred to as genes. So they are parts of DNA only, which is responsible for reflecting different kinds of characteristic. Clear? Different kinds of characteristic. Write down first of all these 3 points. These 3 points. Write down. Chromosomes are bearers of hereditary characteristic. Are bearers of hereditary characteristic and is present inside the nucleus DNA are parts of chromosomes DNA are parts of chromosome only, reflecting different characteristics, not different character. DNA parts of DNA only, chromosomes DNA are parts of chromosome, and is not only confined to nucleus of a cell, and is not only confined to nucleus of a cell, but is present in two other parts. So which are two other parts of the cell where DNA is present? One is mitochondria, and second sorry second, plastids. And since the DNA which is present in mitochondria and plastids is beyond the chromosomes, so they are referred to as what? They are referred to as extra chromosomal DNA. Write down the DNA present in mitochondria, and plastid, mitochondria and plastid, is beyond the chromosomes. DNA present in mitochondria and plastid is beyond the chromosome. As such, beyond the chromosome, they are referred to as extra chromosomal. As such, they are referred to as extra chromosomal DNA, that is present beyond the chromosomes. Extra chromosomal DNA. Clear? Extra chromosomal DNA. And when we talk about genes are nothing but parts of DNA only. They are DNA only, which would be responsible for reflecting different kinds of characteristic. Genes are parts of DNA only, which reflect different characteristic. Genes are parts of DNA only which reflect different characteristics. Which reflect different characteristic. Reflect different characteristic. Clear? So that's how we define chromosome, DNA, and genes. But let's see. Advancement in the field of biotechnology started when? See, advancement in the field of biotechnology started after the double helix structure of DNA was given. And the double helix structure of DNA was given by whom? It was given by Watson and Crick. So Watson and Crick, sorry. The double helix structure of DNA was given by Watson and Crick, and it was given in 1953. After which we can see advancement after which advancement in the field of biotechnology started. Let's draw it bigger because it won't be able to understand it. Let's draw it bigger. Now A stands for what? See, this is suppose the double helix structure of DNA. Now this structure is called as what? Now this was given by Watson and Crick. See, now there are four kinds of bases here. A is a base. A stands for adenine. T stands for thymine. C stands for cytosine. G stands for guanine. Clear? ATCG. Now, A is combining with T. Now, this would be called as what? This would be referred to as base pair. CG also would be called as base pair. Now, there are phosphate units. Suppose this green is the phosphate unit. And in between two phosphate unit is a sugar unit. Sugar unit. So in between two phosphate unit is a sugar unit. And we can see this is the base pair. The joining of these two would be done by hydrogen bond. So that's how the double helix structure of DNA looks like. So that is the double helix structure of DNA. And it consists of base pair, that is AT or CG. And the bases are joined by hydrogen bond. Then it consists of what? It consists of sugar unit. In between the two sugar unit is a phosphate unit, remember? And this was given by Watson and Crick. But we can see that when we are talking about the double helix structure of DNA, with the help of this double helix structure, we are responsible for determining the genetic makeup of organism. Genetic makeup of organism. Just copy this and then we move forward. Genetic makeup of organism. Finished? Take a click or screenshot of it also, so that we can save time. Nothing in this. Fine. Now this is the double helix structure. Now see, when we talk about genetic makeup, there is a map which is called as HEP map. Suppose this is a map which is called as HEP map. This HEP map is responsible for comparing the genetic makeup of different organisms. Genetic makeup of different organisms. So we differ from other organism on the basis of genetic makeup. So what is the genetic makeup? What do we study in genetic makeup? So in genetic makeup of any organism, we study what? We study not only the genes and their variants, but we study the sequencing of base pair. The sequencing of base pair. Clear? We are responsible for studying the sequencing of base pair. And suppose one sequence may be AT, CG, AT. Another sequence can be AT, CG, AT, CG. Another sequence can be CG, CG, AT, CG. So different kinds of sequence which is possible. So you can see each and every organism would be having a separate sequence of base pair. Each and every organism would be having a different sequence of base pair. So when we study the sequence of base pair, we examine the genetic makeup of an organism. This would be called as what? This would be called as genome. So in the year 2000, we were responsible for studying the human genome, that how the genetic makeup of humans looks like. So we were responsible for studying the human genome, and it was deciphered for the first time. Deciphered for the first time, and that was in the year 2000. We were responsible for deciphering the human genome for the first time in the year 2000. But then the question is that what we are responsible for studying in human genome, what we look into. So when you talk about human Human genome or genetic makeup of human is called as what? It is called as blueprint of life. Human genome or genetic makeup of human is called as what? It is called as book of life. So it is called as blueprint of life. It is called as book of life. It includes what? It includes genes, their variants, and some regulatory genes which are responsible for controlling other genes. So in this, we are not only responsible for studying the genes, their variants, but we are also responsible for studying regulatory genes which are responsible for controlling other genes. So that's called as human genome. That's how we define the human genome. But human genome was deciphered in the year 2000 by the scientists of Europe and a private company which is called as Celera Genomics. So it was deciphered in the year 2000 by a company called Celera Genomics and also European scientists. And first of all, we came to the conclusion that earlier the perception was that there are 1 lakh genes in human cell. Now this perception was changed because today we think that only 20,000 genes are present in human cell and not 1 lakh as thought earlier. So this was first change that we noticed. And also we can see that as far as human genome is concerned, in human genome, we studied the sequencing of base pairs for humans, that how in the human cell, in the chromosome of human, in the DNA of human, the sequencing of base pair has been arranged, or sequencing of base pair is there. Now this sequencing is also determined in the case of DNA fingerprinting. So what is the difference between human genome and DNA fingerprinting? Difference between human genome and DNA fingerprinting. But look, as far as sequencing of base pair is concerned, in case of human genome, these sequencing, in case of some sequence of base pairs would be repeated at, first of all, write down, otherwise you won't be able to understand. Write down human genome. Human genome, because if we go too long, then some people may have problem who are not from biology side. And let me see the questions, if any you have asked. Not at all. Good. Write down human genome. Write down human genome. Human genome is the genetic makeup of humans. Human genome is the genetic makeup of humans. It is called the book of life. It is called the book of life, or blueprint of life. It is called book of life or blueprint of life. Blueprint of life. Book of life or blueprint of life. It includes genes, that is, we are talking about the human genes. It includes genes, their variants, genes, their variants, and regulatory genes, which it includes genes, their variants, and also regulatory genes, which are responsible for controlling other genes, and also regulatory genes which are responsible for controlling other genes. responsible for controlling other genes. Regulatory genes which are responsible for controlling other genes. And we can see that human genome was deciphered in the year 2000 by the scientists of Europe and a private company called Celera Genomics. It was deciphered in the year. It was deciphered in the year 2000. It was human genome was deciphered in the year 2000. Clear? And the first conclusion which was drawn was that the cell of human consists of 20,000 genes and not 1 lakh as thought earlier. And not 1 lakh as thought earlier. Clear? 1 lakh as thought. Then that was the first conclusion drawn. But also, the objective of human genome was what? The objective of human genome was to determine the place of humans on the HAP map. The objective of human genome was not only this, but there were other objectives too. Let's look into those objectives. First was to determine the place of humans on the HAP map, that humans are present where on the HAP map, who are their close door neighbors. The animal which is very close to us is chimpanzees. Clear? Because the genome of humans and chimpanzees has very less difference. Clear? Second purpose was that there are diseases associated with the genetic makeup of humans. So the cure for all those diseases can be made. So that's the second purpose. And if we know the genetic makeup of humans, we can also know the genetic makeup of other organisms as such. Genetic makeup of other organism. Now this would be helping us where? It would be helping us in organ transplant. Clear? So there were three main objectives. Write down. There were three main objectives of human genome. There were three main objectives of human genome. Objectives of human genome. First, three main objectives of human genome. First, the purpose was to determine the place of humans, on the HAP map. The purpose was to determine the place of humans on the HAP map. That's one objective. And we can see there are diseases associated with the genetic makeup of humans. So a remedy for these diseases can only be found. A remedy for these diseases can only be found if human genome is deciphered. Write down. There are diseases associated with genetic makeup of humans. There are diseases associated with genetic makeup of humans. A remedy for these diseases. A remedy for these diseases can only be found. A remedy for these diseases can only be found if human genome is deciphered. And the third thing was what? The third thing was that it would be also responsible for helping us in organ transplant. Write down. It would also help us in organ transplant. It would also help us in organ transplant. Here, let's come back face to face and discuss other things also. Now that's about human genome that you have to know. See, another thing that you have to know here is that when human genome was deciphered, first thing was what? First thing was that the we concluded that in human cell only 20,000 genes are present and not 1 lakh as thought earlier. That was the first conclusion drawn. The second thing was what? That in human genome, we were responsible for studying what? We were responsible for studying the sequencing of base pairs. This sequencing we also study in case of what? In case of DNA fingerprinting. So what is the difference between human genome and DNA fingerprinting? Now, the answer to that is what? DNA fingerprinting is what? The unique identification of an individual. The same base pair sequencing is studied. So, what is the difference between human genome and DNA fingerprinting? Now, the answer to that is what? That more than 99% of genes among us is exactly the same. Me and you, more than 99% of genes among us is exactly the same. The difference is only of less than 1%. Suppose if the sequencing of base pair is conducted for more than 99% of the genes which is common among us, this would be called as human genome. And if suppose the sequencing is conducted for less than 1% of the gene which is uncommon among us, now this is called as what? This would be called as DNA fingerprinting. Clear? But then where to determine that how DNA fingerprinting can be conducted and how human genome would be conducted? Clear? So we can see that there should be some difference among us. Where is that difference? We have said that if suppose sequencing of base pair is determined for more than 99% of the genes, which is common among us, then it would be called as human genome. And if the sequencing of base pair is determined for less than 1% of the gene which is uncommon among us, then this would be called as what? This would be referred to as DNA fingerprinting. DNA fingerprinting. Clear? But then how to conduct DNA fingerprinting? How to know that what is the difference between us? Difference between us. There should be some. So if suppose we examine the human genome, the entire human genome, because it took roughly about 7,40,000 A4 sheets to entirely print the human genome. So if we determine the human genome, the entire human genome, we can see that in between there are, as far as human genome is concerned, in between there are certain sequencing of base pairs which would be repeated at frequent intervals. First, then again it would be repeated, then the same sequencing would be repeated. This sequencing of base pair which is frequently repeated is called as what? It is called as short tandem repeat. It's referred to as what? It is referred to as short tandem repeat. And it is the short tandem repeat where we are responsible for differing from each other, where we differ from each other. Clear? So it is the short tandem repeat where we differ from each other. Clear? So this needs to be known to us. Right on. When human genome was deciphered, when human genome was deciphered, human genome was deciphered. When human genome was deciphered, when human genome was deciphered, human genome was deciphered, it was concluded that it was concluded that it was concluded that it was concluded that human cell have 1 lakh, have 20,000 genes, 20,000 genes, 20,000 genes, but not 1 lakh as thought earlier, but not 1 lakh, but not 1 lakh as thought earlier, as thought earlier. Thought earlier. Clear. So human cell is having 20,000 genes and not 1 lakh as thought earlier. So that's one thing. Now, second thing is what? Second thing that you have to know is that in human genome, we were responsible for examining the sequencing of base pair, which is also done in case of DNA fingerprinting. Right down in human genome, we were responsible for examining, the sequencing of base pair. Sequencing of base pair. In human genome, we were responsible for examining the sequencing of base pair, which is also done in case of DNA fingerprinting. So then what is the difference between DNA fingerprinting and human genome, which is also done in case of DNA fingerprinting? The difference is that since more than 99% of genes, among us is exactly the same. More than 99% of genes among us is exactly the same. More than 99% of genes among us is exactly the same. When sequencing is conducted for these genes, it would be referred to as human genome. But if sequencing is conducted for less than 1% of gene uncommon among us, less than 1% of gene uncommon among us, 1% of gene uncommon among us, it is referred to as DNA fingerprinting. So if you conduct If you are responsible for examining, sequencing for more than 99% of the gene which is common among us, it would be called as human genome or genetic makeup of humans. And if we are responsible for examining the sequencing of less than 1% of the gene which is uncommon among us, because gene is what? Gene is nothing but DNA. Clear? Less than 1% of the gene which is uncommon among us, then it would be called as DNA fingerprinting. Clear? DNA fingerprinting. But then how to examine how DNA fingerprinting should be conducted, and where is the place that we differ? Suppose this is the human genome. Took 7,24,000 A4 sheets to print it. We can see in human genome there are some sequence of base pairs which would be repeated after frequent intervals. There are some sequence of base pairs which are constantly repeated after frequent intervals. This sequence of base pair is called as what? It is called as STR. That is short tandem repeats. This is called as STR, short tandem repeat, where we differ from each other, where we are responsible for differing from each other. Short tandem repeats where we differ from each other. Clear? Short tandem repeats. Let's see. When DNA fingerprinting is conducted, so that's the difference between DNA fingerprinting, and the main difference is what? Main difference is STR. Now, when DNA fingerprinting is conducted, how? Suppose a person wants his DNA fingerprint to be conducted. So he would be visiting, suppose, a forensic lab where DNA fingerprinting is conducted. Clear? And sample would be taken from that person. Sample means that samples can be blood, it can be hair with roots. So sample can be taken from that person. So suppose we are talking about DNA fingerprinting. And we know that DNA fingerprinting is the unique identification of an individual. DNA fingerprinting, the method was given by Alec Jaffrey. So this was given by Alec Jaffrey. Fine. DNA fingerprinting, the method was given by Alec Jaffrey. And this is unique identification of an individual. How it is conducted? First of all, you go to the lab where DNA fingerprinting is conducted, they would be asking you sample. And sample is what? Sample is blood. Clear? And remember that in blood, only WBC has DNA, not the RBC. WBC has DNA, not the RBC. Clear? So first of all, we will be responsible for taking the sample, and from the sample we are responsible for extracting the genetic material. So suppose this is the genetic material which has been extracted from the sample. And then we will be cutting a portion of DNA. So we are responsible for cutting a portion of DNA. And which portion we would be cutting? We would be cutting STR only. We would be responsible for cutting a portion of DNA. And this would be called as what? This would be referred to as short tandem repeats. You know that. This portion would be cut. Clear? This portion would be cut. And then we would be conducting And we would be responsible for conducting what? We would be responsible for conducting DNA fingerprinting on it through a test, and that short tandem repeat, and then we would be conducting a test which is called as DNA fingerprinting. So DNA fingerprinting would be conducted here, and as far as short tandem repeat, so DNA fingerprinting would be conducted, and we can see as far as DNA fingerprinting is concerned, from first of all, we would be conducting a test here which is called as southern blotting test. Southern blotting test. Southern blotting test. And in southern blotting test, what we do is we use liquid. And this liquid is called as what? It is called as alkaline phosphatase. This liquid is called as alkaline phosphatase, in which the sample that is called as short tandem repeat is used there. And after this test is conducted, we can see the sequencing of base pair would be visible. And this sequencing of base pair which is visible, we can confirm that this is DNA fingerprinting now. DNA fingerprinting has been confirmed. DNA fingerprinting would be now confirmed. And we can see that this test is called as what? This test. I repeat once more. First of all, what we are doing is we are responsible for taking a sample, biological sample, which can be blood. And how can we take blood? So we are taking biological sample blood, and in this we would be seeing that only WBC has DNA, not the RBC. WBC has DNA, but RBC does not has DNA. No DNA is present in RBC. So what we would be doing? That first of all, the portion of DNA is cut, and the portion of DNA is cut with the help of restriction enzyme. And which portion would be cut? The portion which is cut is called as STR, where we differ from each other. Clear? So this portion is cut with the help of restriction enzyme. And restriction enzyme, and then on this STR, short tandem repeats, that is the part of the DNA which is cut with the help of a restriction enzyme, we are conducting a test which is called as southern blotting test. So southern blotting test would be conducted on this. So when southern blotting test is conducted, then we can see the sequencing of base pair would be revealed. So in this test, we use a liquid which is called as alkaline phosphatase. So a liquid is used, and this alkaline phosphatase would be used, and then the southern blotting test is conducted. Clear? So southern blotting test is conducted. Fine. So this is how it is conducted, and after the southern blotting test, we can see the sequencing of base pair is revealed. But this is not revealed directly. You have to pass it through X-ray, then only it can be revealed. Then this method is called as DNA fingerprinting. Write down, please. DNA fingerprinting. Sir is Uh, genes are the same number for adults and child. No, they are same. Just write down one DNA fingerprinting and then we end the class. Clear? DNA fingerprinting. Write down and then we end the class. DNA fingerprinting. DNA fingerprinting is the unique identification. Is the unique identification. Identification of an individual. Unique identification of an individual. And we can see the purpose of conducting DNA fingerprinting is not only to identify a person, it can be used for identifying a dead person. It can be used for identifying not only a dead person, but also a criminal. Write down. The objective is to not only identify. The objective is not only to identify. Not only to identify biological parents. The objective is not only to identify biological parents, but also criminals, and dead. So dead person can also be identified. For this purpose, biological sample is required. For this purpose, biological sample is required, which can be blood. There are many samples here with roots, bloods. So all these can be used. Which can be blood. But do remember that in blood, only WBC has DNA, not the RBC. Can be blood. And first of all, a portion of DNA is cut, and you know which portion. First of all, a portion of DNA is cut because it is, and in bracket write down STR, that is short tandem repeats. A portion of DNA is cut, that is short tandem repeat. Let's complete this portion, this topic, and then end the class. Short tandem repeat, portion of DNA is cut, short tandem repeat. With the help of restriction enzyme, with the help of restriction enzyme, which is also referred to as molecular seizures, which is also referred to as molecular seizure. Which is also called as molecular seizures. Here, and on this sample, we can see that a test is conducted, which is called as SUL. So when you cut it, at that time the sequencing of base pair is not visible. On this sample, a test is conducted. On this sample, a test is conducted called Southern blotting test. A test is conducted called Southern blotting test. Southern blotting test. Clear? In which the liquid used is, alkaline phosphatase. So we are using for Southern blotting test a liquid, in which the liquid, now this is the liquid where the sample would be immersed, in which the liquid used is alkaline phosphatase. After this test, a purple precipitate is visible. After this test, a purple precipitate is visible. Precipitate is visible, which is passed through X-ray, which is passed through X-ray to reveal the sequencing of base pair, which is passed through X-ray to reveal the sequencing of base pair. Reveal the sequencing of base pair. Clear the sequencing of base pair. So this is how it is conducted. But tomorrow we are going to study that how DNA fingerprinting would be having exceptions, that whether a person can have more than one DNA fingerprint. What happened was that in US, a paternity test was conducted. A paternity test was conducted, and paternity test, you understand that whether the person is the father of his own son or not. So paternity test was conducted on a US citizen. What happened was, honestly, he was the father of his own son, but he failed that test. Don't laugh. And he failed that test, and it was found that the child belongs to his uncle. And don't think mischievous about it. No illicit relationships. So child was found to be his uncle. But the next line which I say would be surprising you. The uncle was never born. So what is this we are talking about? We are talking about a condition in which a person can have more than one DNA fingerprint. Tomorrow we'll discuss that. So thank you so much. And let me see that if suppose you have any query right now. So we'll take up an interesting topic tomorrow. Now the class is over, because midway we cannot leave the topic. Okay, thank you so much. Thank you so much.